B2.1 Osmosis and Plant Transport. So the definition for osmosis. Osmosis is the diffusion of water molecules from a dilute solution, like the diagram there, the diagram on the left where there's lots of water molecules, to a more concentrated solution, the diagram on the right where there are less water molecules, through a selectively or partially permeable membrane. So the key thing to know is that it is the movement of water, 1 mark, from a dilute to a more concentrated solution, for the 2nd mark, through a selectively permeable membrane, for the 3rd mark. So an example that we could look at here of osmosis is placing plant tissue into different sugar solutions. So we have a dilute sugar solution on the left and a more concentrated sugar solution on the right. The length and the mass is taken at the beginning, and then 4 hours later the length and the mass is taken again. If we look at the solution on the left, the dilute sugar solution, we can see that after 4 hours the length and the mass have both increased. So if you were being asked to explain why, we can say that they have increased in mass as water has moved in by osmosis, for 1 mark, through a selectively permeable membrane, for another mark, from a dilute solution in the beaker into the plant cells, which is a more dilute or more concentrated solution, for the final mark. Looking at the diagram on the right, in the concentrated sugar solution, we can see that the length and the mass have both decreased. So to explain that, we can say that water has left the plant cell by osmosis through a selectively permeable membrane, moving from the more dilute solution, which was inside the plant cells this time, to the more concentrated solution in the beaker. So always look at how many marks are being awarded. Make sure you've mentioned the term osmosis, which is worth a mark, what direction the water is moving, so where is more dilute and where is more concentrated, and that it's passing through a selectively permeable membrane. You could also be asked to work out the percentage change in length or in mass. So to work out the percentage change, you need to know the change, the difference, so the before and after, the difference, and then you always place it over the initial length or mass, or whatever the question is asking you about. So for example, if you were being asked about the percentage change in mass for this dilute solution, so it went from 1.03 to 1.57, so that is a change in mass of 0.54, divided by the initial mass, which was 1.03, multiplied by 100%, and that would tell you that the cell increased by 52.4%. Another practical could be where we have placed different lengths of potatoes in different solutions of sugar and leave for a period of time, and you can either measure the length or the mass. So the graph on the right here is showing you the change in mass, percentage change in mass, which again would be calculated by the change in mass over the initial mass. And where it crosses the axes, that is what the point is when you would say that the inside of the cell is the same as the solution outside of it. So the concentration is equal inside the potato to the concentration of the solution outside. And on this graph, it would be reading around 0.41 moles per decimeter cubed. Osmosis in plant cells. So our typical plant cell, a normal cell is shown in the second one over. So you may be seeing it in being placed in a dilute solution or being placed in a concentrated solution. So if we look at the diagram on the left, when it's placed in a dilute solution, we can see that the vacuole has increased in size because water has moved in through the selectively permeable membrane by osmosis, and the cytoplasm has pushed the membrane against the cell wall, and it has made the plant cell turgid. The diagrams on the right show us that it has been placed in a more concentrated solution, so the vacuole has decreased in size. Water has left the plant cell by osmosis and entered the solution, so the cell has initially become flaccid, and then over time, as it loses more water, the cell membrane starts to pull away from the cell wall, and we say that the cell has become plasmolysed. The cell wall will prevent the cell from bursting, which is different to our animal cells. So osmosis in animal cells, if we placed, for example, red blood cells into a very concentrated solution, the water will move out of the cell into the solution. So it will move by osmosis out of the cell by osmosis through the semi-permeable membrane or selectively permeable membrane, and we say that the cell has crenated. If we have an equal concentration, then the water potential, so the concentration of water inside and outside the cell, is the same. So there is no net movement of the water and the cell is normal. If we place it then into a dilute solution or into pure water, water will move into the cell by osmosis because there's more water in the solution than there is in the cell. This will cause the cells to swell, and because they have no cell wall, they will burst, and this is known as lysis. So cell lysis is then bursting. So what do plants need water for? So plants need water for 4 things. They need it for photosynthesis. So here's your word equation for photosynthesis. The carbon dioxide plus water in the presence of light and chlorophyll gives glucose plus oxygen. They also need it for support. So as we've looked at there, water will enter the vacuole, make the cells turgid. So you can see the cell on the left turgid compared to the plasmolysed cell where the cytoplasm has pulled away from the cell wall. Also need it for transport, so minerals will dissolve into the water and be transported through the xylem. And then the 4th one is transpiration, where water is lost by evaporation. So we're going to look at that in the next slide. So transpiration is a movement of water. So this is part of the leaf. This is showing us the spongy mesophyll cells, so the lower part of the leaf, the guard cells in the stomata. So when you're describing how water is lost by transpiration, the water moves from the xylem into the spongy mesophyll cells. It then evaporates from the surface of the mesophyll cells and moves into the intercellular air spaces in the spongy layer. And then it will diffuse out of the stomata when there is a concentration gradient. So that would mean that there's more water molecules inside in the air spaces compared to the surrounding atmosphere. So the water moves by diffusion out through the stomata. It is not osmosis because it is not passing through a cell membrane or a partially permeable membrane. So transpiration, the evaporation of water molecules from the surface of the mesophyll cells, and then the water diffuses out through the stomata. Ways that rates of transpiration can be measured. The first one here is a bubble potometer. So you can change conditions such as light intensity, air movement and temperature. And then using this apparatus, you can record how far the air bubble moves over a period of time. Or you could also measure the time it takes for an air bubble to move a set distance. But the 2 things you do need to be recording to work out rate are time and distance. The reservoir in the diagram would be used to refill the water and put the air bubble back to 0 to allow the experiment to be done again. A mass spectrometer is similar to this, where you would weigh the apparatus, leave it for a period of time in the conditions you want to change, and then take the new mass. And again, you can work out the rate of transpiration because you have the change in mass and you also have the time. So the factors that affect transpiration: temperature, humidity, air movement, and light intensity. So light intensity, so if you increase the amount of light or the light intensity, transpiration will increase. This is because the stomata will open wider to allow more carbon dioxide to enter the leaf by photosynthesis, and because the stomata are open, then more water can evaporate and diffuse out through them. Temperature. As we increase the temperature, transpiration increases. This is because the water particles will gain more energy, kinetic energy, so evaporation and diffusion will be faster at higher temperatures. If there is wind or air movement, then transpiration will be faster in the windy conditions. Again, this is because the water vapour is removed from around the stomata, helping to maintain a concentration gradient between the inside of the leaf and the outside of the leaf. So more diffusion will happen for the water diffusing out through the stomata from the higher concentration inside the intercellular air spaces to the surrounding air. Humidity is the only one that will decrease it. So if you increase the amount of humidity, so the amount of water in the atmosphere, then transpiration will decrease. And this is because there's a lower gradient, concentration gradient. There's less of a gradient or difference between the inside of the leaf and the outside of the leaf, so diffusion will slow down. So here are some example questions that you could be asked about transpiration. So for foundation tier, Fiona was asked to carry out an investigation in the lab to compare the rate of water loss from the upper surface of one leaf with the rate of water loss from the lower surface in the second identical leaf. The diagram shows the apparatus and materials that she was given. Describe how Fiona would set up and carry out her investigation. So this is a 6-marker question. You can see that the word rate has been put in bold, so you need to be thinking how you can measure rate. So if you can pause the clip and see if you can answer this for 6 marks. So within your answer, you need to ensure that you are explaining how each part of apparatus is used in this investigation. So you would be taking the 2 leaves. One of them will have the upper surface covered and one will have the lower surface covered with a petroleum jelly. You would weigh the leaves before and after a period of time. You would then place one leaf, as I said, with the petroleum jelly on the lower surface. You would weigh the leaves before the experiment and record the results. You then would paperclip the leaves, so telling what you use the paperclip for, to the string that is attached to the retort stand. You leave it for a period of time, because remember, time is needed to work out the rate, and you measure the time using the stopwatch. And you need to state that because the apparatus has been shown. And then after a period of time, for example an hour, you would reweigh the leaves and you would calculate the change in mass, and then you can work out the rate by the mass lost, the change in mass divided by the time. The leaf that loses the most mass would indicate the surface that loses more water. So in this investigation, if you were comparing the rate of water loss from the upper surface of the leaf to the rate of water loss from the lower surface, you would expect the lower surface to lose more water because it has more stomata. Name 2 factors other than humidity that Fiona would need to control during her investigation. Well, other things that will affect the rate of transpiration will be temperature or air movement. Name the pores in the leaves through which water is lost. Stomata. Which of the graphs A, B or C show the rate of water loss if the humidity around a plant increases? So if humidity increases, there's more water in the atmosphere. So that means that the rate of transpiration decreases. So the graph would be graph B. And give 2 uses of water in plants. Well, we've given you 4 earlier. So you could say photosynthesis, support, transport. We've already done transpiration within the question. But these are the general ones that they look for. Transpiration, photosynthesis, support, turgid, transport, cooling or solvent. A higher tier question, again, starting off with give 2 uses of water in plants. So the same thing, transpiration, photosynthesis, support, turgid, transport, cooling or solvent. So students used a photometer to investigate how different treatments of the leaves affected the rate of water uptake in plant shoots. The diagram shows a photometer. Name another piece of apparatus needed by the students to work out the rate of water uptake in the plant shoot. Well, to work out rate, we also need to measure the time, so you would need a stop clock or a stopwatch. The students carried out the investigation using 4 identical plant shoots and Vaseline. Vaseline is a waterproof jelly that blocks stomata. The table below shows the treatments given to the plant shoots. So A had no Vaseline on any surface, B the upper surface was covered, C the lower surface is covered, and D the upper and lower surfaces were covered. And all other conditions were kept constant. So the students measured the rate of water uptake for each plant shoot, and the graph shows the results for B, C and D. So on the graph, draw the bar that you would expect for plant shoot A. So plant shoot A had no Vaseline on it, on either surface, so you would expect more water vapour to be lost from it compared to plant shoots B, C or D. So the bar being taller than B would be what would be expected. Each plant shoot was left for 5 minutes in the photometer before readings were taken. Suggest why. So for that, you would be thinking, and the idea is that it allows the time for it to settle, to acclimatise to the conditions you're setting up in, to reach a steady rate, to allow time for the bubble to start to move. The plant shoots have stomata on both the upper and the lower surfaces of the leaves. Use the graph and the table and the graph to describe and explain the difference in the results for the rate of water uptake in plant shoots B and C. So this is a 3-mark question. Describe, say what you see, and then explain the difference. So we can see that B had only the upper leaf surface covered. C, the lower leaf surface was covered. And when we look back at the graph, we can see that B had a greater rate of water uptake than C. So why would that be? So more water uptake in B than C is your description. Explaining why. Well, if the upper surface is covered, that means the lower surface is exposed. The lower surface has more stomata, which is what your first marking point is. Therefore, more water loss, more evaporation, more transpiration, more diffusion is happening in B, or less diffusion is happening in C. The students then placed a clear plastic bag over another plant shoot. They worked out the rate of water uptake for this plant shoot, and the diagram shows the setup. The rate of water uptake in this shoot was different compared to an identical plant shoot not covered with a plastic bag. Describe and explain the difference in the results. So the plastic bag would make the conditions inside more humid. So if it is more humid, there's going to be more water vapour within the atmosphere surrounding the plant. So we would expect the bubble to not move as much. So the rate of uptake would decrease because of that. So the higher humidity in the bag, or water builds up in the bag, therefore there's less water loss or less transpiration, less evaporation, less diffusion, or less gradient for water loss.